Good morning everyone, my name is Sinobert Indao and I'm here to report all about genetic engineering. Objectives. After going through this module, you are expected to number 1. Define genetic engineering. Number 2. Describe the techniques in genetic engineering as based from the situation. Number 3. Appreciate the role of genetic engineering to human life. And hybridization. When two individuals with unlike characteristics are crossed to product the best in both organisms, like the disease resistance potato called the Bourbon potato. Second, inbreeding. A technique of breeding organisms that are genetically similar to maintain desired traits found in the pure dog breeds. Breeding closely related organisms to maintain specific traits such as purebred animals. Third, artificial selection to indirectly manipulate genes focusing on the physical trait among organisms, breeding for disease traits without control of what genes can be passed. Selective breeding, maintain animals or plants with specific traits to enhance those traits in offspring. Selective breeding, a process when animals with desired characteristics are mated to product offspring with those desired traits, such as Angus cows, and breed to increase more meat. To maintain selective breeding, maintain animals or plants with specific traits to enhance those traits in offspring. What is genetic engineering? The direct manipulation of an organism's genes using biotechnology involves different kinds of technologies used to alter the genomes, that includes inserting genes from same or different species, aims to product or improve product. Example, imagine giving a plant a gene from a bacteria to help it resist pests.
I'm Gerald Bongo. I'm reporting the science. First, definition of genetic engineering is the process of changing the DNA in living organisms to create something new. Involves modification, recombination, or manipulation of DNA or RNA. So, ang genetic engineering mao na siya ang proseso sa pag-usab sa DNA sa mga buhi nga organismo aron makahimo og bagong butang. Next, recombinant DNA technology. Recombinant DNA is made by mixing DNA from two different sources. First, Swiss microbiologist Werner Arby discovered resection enzymes in 1968 used to splice, connect, or ligate, and remove or add nucleotides to sequences of the DNA. So, ang recombinant DNA technology mao na siya ang proseso sa paghimo og DNA pinaagi sa pagsagol sa DNA gikan sa duha ka lain-laing tinubdan. Process. This process is used in recombinant DNA technology to remove and insert genetic sequences from and into other sequences of other organisms. Inserting the desired gene into the genome of the host involves the selection of the desired gene for administration into the host, followed by a selection of the perfect vector, either plasmid or bacteriophage, with which the gene has to be integrated and a recombinant DNA is formed. So, ang process sa recombinant DNA technology mao ni siyang magamit sa pamaagi aron kuhawon og ibutang ang genetic sequences gikan sa usa ka organismo padung sa lain. So, next. Last, process. The recombinant DNA has to be introduced into the host, maintained and carried forward to the offspring. Biotechnology and pharmaceutical companies practice these techniques in working with medical and research purpose. So, ang process nagpasabot nga ang recombinant DNA kinahanglan isulod sa host ang pingan o ipabilin o ipasa ngadto sa anak or offspring. Thank you.
show how the woman growth hormone genes is inserted bacteria cells production. Nagpakita kung giunsa pagbutang ang woman growth hormone genes sa bacteria. Una, gikuha ang genes gikan sa tao unya gisagol sa DNA sa bacteria. Human ibutang ubalik sa sulod sa bacteria aron makaproduce og woman growth hormone genes nya gamit sa pagtambal sa mga tawo nga kulang sa hormone genes. For easy understanding of process recombinant DNA, 1. Select of the desired genes. 2. Choose a vector, plasmid, or bacteria's page. 3. Insert the gene into the vector. 4. Introduce into the host organism. 5. Genes is maintained passed to offspring.